bit of introduction about this session. So ROKIT has been doing this with some other institutions also. So this is the journal club slash webinar about bioprinting, focusing on specific research topics. Oh, okay.
Yes.
Specific on research topics. They have done this with Cleveland, our very own Cleveland Clinic. They have done this with, I think,
UIC.
UIC. I think you have done this with Stanford or MIT. I'm not sure about
Yeah, we
that.
have done with Stanford as well.
Okay, so I think when we were talking, Senthil said, why don't we do this webinar or slash the seminar thing with CSU also, and I was interested, and that's how I reached out to Chandra and Muyal, and that's how we have the others also. So yeah, I won't speak
So
anymore.
actually,
Dai, on to you.
you made the introduction. Most of what I wanted to say, you have already said. So that's pretty easy for me to
Okay.
then go forward. So again, good morning to every one of you. I am Senthil. I am the founder and CEO for ONS Bio LLC, which is the distribution partner in the Midwest for ROKIT Healthcare. I've been a distribution partner for ROKIT Healthcare for nearly 2.5 years. And I really thank Prabha for giving us this opportunity today to connect with you all and talk about bioprinting and how it's a powerful research tool. And also, we will be touching upon its potential as a therapeutic platform in the context of some emerging mega trends in science and healthcare. And so beyond the basics of bioprinting, we'll also try and cover as many as possible exemplary real-life applications and case studies of both ROKIT's 4D bioprinting technologies as well as maybe other notable bioprinting technologies from both research labs and in some cases also from commercial industries across the world. And also, I will touch upon some of the operating room applications that Rokit's in-house R&D is doing. And as Prabha mentioned, today's talk is like a it's an active outreach to researchers and clinicians across the US that Rokit has embarked on to educate and create awareness around this potential for bioprinting technology to transform, impact, and advance both research as well as generation of regenerative therapies. ROKIT Healthcare, about the company itself, is a South Korean pioneer and it's a leader in this field. It's a well-rounded regenerative medicine company. It goes beyond just manufacturing bioprinters. Notably, ROKIT Healthcare is the manufacturer of 4D bioprinters. We don't like to call it 3D, and Dai will explain why. Um, the 4D bioprinters and organ regeneration platform technologies that is called in vivo 4D and 4D6. Again, it has a lot of potential and applications in both basic research and translational research, as well as in the development of autologous cell therapies. And it's also worth mentioning that ROKIT manufactures industrial 3D printers as well. So their engagement with this additive manufacturing goes way beyond bioprinting. And they also offer genomics and single cell RNA services, among other things. Though some of these offerings may not be yet ready for the US market. And about Dai is a wonderful colleague and very knowledgeable expert in bioprinting and the in vivo platform. She's been with ROKIT Healthcare for quite some years now, and she'll say how many. And she's a senior bioprinting consultant and specialist, and she's also the global business development manager. And she's a very passionate advocate of bioprinting technology. And she will now give us her talk. Dai, over to you.
Yeah, thank you, Senthil. Hi, everybody. My name is Daye Lee. As Senthil introduced, I am a senior bioconsultant at ROKIT Healthcare. I've been here for about 3.5 years now, and I'm very used to speaking at this late night hour in Korea to accommodate the US time because I am a manager for that territory and have been working with Senthil for the last several years as well. So I'm very fortunate to be here, and thank you so much for your invitation to the Cleveland State University group meeting. Um, to introduce myself a bit, before joining ROKIT Healthcare, I was a research associate at the MIT Koch Institute of Integrative Cancer Research. And one of the things that I was doing there as a research associate was studying interactions of the immune cells with lung tumors using the mouse models. And there was a word that was always sort of at the center of our discussions among our groups, and that was called the TME, which you guys may be familiar with as the tumor microenvironment. And the idea was always that if we could better, map and visualize and manipulate this three-dimensional tumor microenvironment that is dictating cancer behavior, that would really be the first step toward understanding cancer tissues in general, and also taking the first bold step toward the holy grail of therapy. And that's really what got me personally interested in the technology of bioprinting using living cells and biomaterials. And so within that context, I would like to share today this session that I've titled On-Demand Tissue Systems Engineering with 4D Bioprinting. And I'm gonna be sharing about the basics of the technology for those of us who may be new to it, but also sort of summarize the benefits or the values that the technology brings that is in contrast with the existing methods of culturing, cell culturing. And also in the end, I will cover some real life applications and case studies of the technologies around the world, taking a look inside the research labs, the commercial industries, as well as in the operating room, with some of the initiatives that ROKIT Healthcare has been leading throughout the world. So if you take a look at this slide, you may have seen this before from a Nature article that visualizes a simulation of how tumors grow over time. And each color you see here on the left and on the right represents a different mutation. And the more successful one of these mutations is at reproducing, the more its color dominates the tumor. And the figure on the right, which shows these bulges in 3 dimensions, further demonstrating how tumor cells or tumor tissues could outgrow, migrate, and squeeze into surrounding healthy tissues. But the point is that these tissues are exhibiting a 3-dimensional environment, and they're exhibiting a 4-dimensional environment because they're changing over time. And so the truth of the matter is that cancer is an extremely heterogeneous tissue, both architecturally as well as spatio-temporally. And in order to model the complexity in its content and dynamic nature of behavior, we must have better tissue models in general. And this is not only true about cancer, as we all know, but is true about human tissues in general. We have a need to spend more time refining the traditional cell culture systems that our understanding of the human body has relied on, because we now understand that 2D cell cultures just do not mimic the characteristics of what happens in vivo. 2D culturing has been the prevailing method, we understand. It's simple, low cost, and highly reproducible. And a lot of the, for example, standard procedures begin with 2D cell culture before moving on to animal models and clinical trials. However, the percentage, as we see in industries, that makes percentage of compounds, novel compounds, for example, that make it to the clinical trials out of the first stage of 2D cell culture is less than 2%. And because of that striking low probability, the research world has been moving on rapidly to the adaptation of 3D models in various research. However, even though we have made some great strides in developing novel 3D models of tissues, existing 3D models still have their own limitations. And if you can take a look at this options at top, these existing 3D models of human tissues include freeze drying, solvent casting, electrospinning, spheroids, and the use of decellularized animal tissues to create the three-dimensional environment to seed the cells in. And of course, there are those mouse models that we often rely on. However, while a lot of these have been the better methods for mimicking what happens, they have had some limitations, including difficulties in controlling cellular composition and deposition. Some processes like freeze drying and solvent casting may require a lot of pre-processing steps that could potentially have adverse effects on the downstream results. We've got limitations of still having to use some animal-derived components. And finally, for things like the 3D spheroid cultures, there is that intrinsic problem of having necrotic cores in the center without the ability for nutrients to go deeper all the way into the core of the tissues and leaving them hypoxic. And so if you actually take a look at this picture on the right, showing a human skin equivalent model made using the manual casting approach versus the 3D bioprinting approach, you can see the differences here. While the bioprinted one obtained shows a perfectly circular shape through an even distribution of the cells, the manual casting approach has led to a disfigured shape that ultimately resulted in abnormal pigmentation in the center. And so we can understand that just the simplicity of evenly dispensing cells across an area can be very important for reserving the functionality of that output human tissue. So it is necessary to engineer more powerful and reliable in vitro 3D models for better dissecting the roles of various physical factors, biochemical cues, and microenvironmental elements that directly impact the tissue behavior, progression, and its responses to perhaps drugs. And for these reasons, bioprinting has been rising to attention as sort of the next level of fabrication. Many of you may be familiar, but for those of you who are new to it, if we were to define bioprinting, I would say it's just a perfect combination, the best of both worlds between computer science and biology. So according to a computer-aided design, bioprinting allows you to basically redraw the complex compositions of biomaterials, cell types, and vascular networks that a tissue exists in. And even though early 3D printing technologies were limited to rigid materials such as hard plastics, metal, etc., but bioprinting is different because it offers you a way to fabricate with soft fluidic gels, which are called the bioinks. And these gels are often combined with cells so that you are essentially creating a living three-dimensional human tissue with living cells, plus the ability to incubate them over time to see their changes in fourth dimension. And that is why we we rely on the word 4D bioprinting rather than 3D bioprinting. I think that is a more apt description of this technology. And so if we can create these better models, we must understand that bioprinting is also a platform technology. It's not limited to just some types of cells or some types of tissues or biomaterials, and it can really have implications across a wide variety of themes and industries. And so eventually, these bioprinted tissues are going to have a lot of I believe they're going to have only a lot more funding over the years because they can make economic impacts in the biomedical world by addressing some of the age-old problems that we have not been able to solve. And that includes the growing bans on animal testing, the needs for better human physiology and disease models, and the need to save time and cost of R&D in novel drug developments by having these human tissue models that allow you to fail faster before human clinical trials begin. So to summarize, bioprinting technology offers you 3 key values. Number 1 is the architectural complexity using free-form design. And as you can see here, this is an example of creating a human brain cancer tissue models on a chip that includes the inclusion of not only the glioblastoma cells, but also the endothelial cells and a gas permeable channel so that you can mimic a better tumor microenvironment with the vascularization on top of the brain cancer cells. So other than architectural complexity, it brings the value of even automated dispensing. And you have already seen why it is important to have an even dispensing of cells within a tissue, as the cell clustering or the cell distribution can also have impacts on the later outputs. And the key thing is automation. So instead of certain things that we have been doing by hand, now we are able to automate a lot of the processes such as cell dispensing, media dispensing, UV cross-linking of the biomaterial all in one device in order to save our time. And finally, the last but not least biggest value that bioprinting can bring to your work includes the ability to adopt a lot of materials into one construct. Like I said, bioprinting differs from other 3D printing technologies in that it can accommodate not only pellets and plastic filaments, bioplastic filaments, but it could also accommodate hydrogels with or without cells, so that the potential for this application goes to not only things like soft robotics, but also tissue engineering and regenerative medicine. So here I just wanted to show you some snapshots of the things that ROKIT Healthcare's own proprietary bioprinting system offers, with the 6 printheads and up to 384 dispensing features. And that gives us a picture of what the architectural complexity and the even automated dispensing could look like with a bioprinting system. But more importantly, we have been very much focusing on taking this bioprinting technology to the next level, not just focusing on the printing of cells or materials into construct, but focusing on how do we create a chamber environment that allows the printed outputs that you have just created to uphold high cell viability and high functionality even as time goes on. And so if you take a look here, we've, as biologists and life scientists very much understand that because cells are living, they are very sensitive to their physiological conditions, such as temperature, humidity, and CO2 levels. And their cell viabilities and proliferations directly impact how they function as parts of a tissue. And so we have built into our system the ability to control the temperature, the CO2, and humidity of the chamber so that the printed output has an ideal mammalian cell culture environment. And so we call this a bioreactor built-in bioprinter. And so over the past, I would say, 5 to 7 years, there's really been an explosion of the number of publications focusing on finding the ideal combinations between cell types, the biomaterials, and 3D designs in order to create a specific model system for human tissues. And some of these hyperlinked numbers on this slide represent a lot of these review publications that you could refer to for becoming launch pads for your next research. But I just wanted to show here that these publications are all results of researchers who have adopted the bioprinting technology into their labs thanks to the commercialization of the technology to a wider market, and the ability for a lot of these knowledges to be shared on a forum, much like in this seminar. And so I hope that just as this moment as the beginning, we could become a hub for research knowledge sharing and experience sharing for a technology that is relatively novel, but is definitely growing in momentum in the research world. So in the next 10 minutes or so, let me go over what exactly is bioprinting and what are some of the biomaterials that can be accommodated and how does the design of the software design works. And then I will end with some real life applications of the of bioprinting. So when we say bioprinting, it is mainly composed of 3 components. Number 1 is the hardware in the center, the equipment that creates 3-dimensional architectures. Number 2 is the biomaterials on the left. These provide basically in vitro ECM environments for the cells to live in as they are extruded out of nozzles and printed onto in vitro Petri dishes or well plates. And finally, you've got the design software or the slicing software that creates a file that the printer can interpret in order to guide its printing process. In the biomaterials, there are largely 3 categories of biomaterials you could use. One is the naturally derived biomaterials, second is the synthetic derived, and third is the composites. The naturally derived biomaterials include materials like collagen, gelatin, elastin, chitin, etc. that could be coming from mammalian cells, the alginates and the silk from plants, alginates, cellulose from the plants, or from sea organisms, ocean organisms, silk from worms, and hydroxyapatite also from just mammalian bones. These are all naturally derived materials that, that can be sourced from animals, plants, or the oceans. But because of some of the limitations of naturally derived materials, such as the mechanical strength, sometimes synthetic biomaterials are also being used, and these often are biodegradable polymers with enough strength and elasticity with references of use in the US FDA-approved medical devices. And these give you a wider portfolio of biomaterials to use in tissue engineering. But even when these two separate categories of biomaterials cannot meet the requirements for strength, mechanical strength, or biocompatibility, these are often combined to create composites with combined mechanical or biochemical properties to serve as the bioengineered ECM mimics. But the point is that these biomaterials, whether they are soft, low viscosity hydrogels or hard biodegradable plastics, when these are combined with cells for printing, they serve the purpose of protecting the cells from shear pressure as they get extruded out of nozzles. And they also serve the purpose of an engineered extracellular microenvironment for cell proliferation, function, and homeostasis. And these are some of the Bioink properties that we have to be mindful of in order for successful bioprinting processes. And these properties include how printable is the ink? Does it exhibit shear thinning characteristics as it goes through a large amount of pressure through tiny nozzle tips? Does it have viscoelasticity and the ability to actually protect cells from stress? Does it have enough hydration degree for sufficient nutrient diffusion when cells are printed with it? And the other properties include viscosity, biocompatibility, and gelatin kinetics. But the two things that are always in balance is the biocompatibility or the high cell viability versus the printability. Is it able to go through tiny nozzles, and is it able to stack itself up on its own after it's printed in order to preserve the three-dimensional architecture? And so with these sort of things in mind for the biomaterials, how they can be fabricated, what design strategies can be used with the bioprinter would be important in order to optimize the use of these different biomaterials in one construct. Right now, bioprinting in the market includes a variety of methods, actually. The one we are talking about and the one that consists of almost 70% of the market today is extrusion-based, which basically means materials are shot out of dispensers through tiny nozzle tips, either by air or by mechanical motors. And these materials are laden on a flat plate, for example, on a layer by layer basis in order to create a construct, much like what you see in this video on the right. And the strategies are normally, they divide into two broad categories of the scaffold laden versus the scaffold free, where the scaffold laden consists of printing with sort of the harder biomaterials to create a house, and then the seating of cells inside in order to support the living cells architecturally. On the other hand, scaffold-free simply relies on the printing of soft fluidic gels, much like what you see here in this video, with the cells included. And as time goes on, these gels are consumed by either by cells or they're diffused into the nutrient media. And in the end, all you're left with are the cells that have been created into three-dimensional tissue structures. And with the bioprinter, once again, the focus today has been how do we include a chamber system that allows these living tissues printed to actually work the way they should in downstream applications, whether it is for drug screening or for tissue implantation or other things. And so the bioprinters, the key things that bioprinters should include disinfection capabilities, the filtered air, as well as the bioreactor functions. And finally, the third component of bioprinting is the design. When we say design, we are talking about three-dimensional object files, and we're talking about how they get converted into the right G codes or the machine language that dictates how bioprinting, how the hardware works to dispense materials in X, Y, Z directions. As you can imagine, every STL file or every three-dimensional object can have multiple G codes because you can change for example, the speed at which you print. You can change the design of the infill patterns inside an object. You can design how thick you want to make the walls of your construct, etc. And so one STL file can have multiple G-code languages, just like you can see below here with this very simple example of a box. This is a 3D object file being converted into different G-code files of various infill densities. And so it is very important to understand how you optimize the slicing parameters in the software in order to create the optimal quality bioprinted outputs. And so if we put all of these together, the stages or the steps inside the process of bioprinting can be summarized into 5, and that includes the preparation stage with the problem identification. What do you want to solve that traditional cell culturing techniques have not been able to solve? It includes material selection, the cell selection, and 3D modeling. Then the second step is the printing, the loading of the materials, the optimization of the settings with which you will create the G-codes from the same STL file. And then it is followed by the actual fabrication process with your hardware. The third step is post-printing, and this is often required. This is almost always required with if you're printing with soft fluidic gels that need some cross-linking before they can go into the cell incubator. We want to make sure that they don't just spread all over when they actually get submerged in media. And so the post-printing process will include cross-linking, which means you either increase or decrease temperature to make your construct solid. You can also introduce novel chemical or ion solutions to solidify your construct, much like when you need to add calcium or barium ions into alginate in order for it to solidify. Or you could introduce light, whether it's UV light or visible light, to your construct so that it cross-links. The post-printing process also includes what happens inside the cell incubator, including differentiation if you are working with stem cells. It includes, of course, the general proliferation stages that cells would go through. And finally, if you are studying something like the skin, trying to create, the epithelium, you may want to print on top of wells that have the air-liquid interface instead of just regular plates, and just create the right environment for those 3D printed cells to live in. Finally, the fourth and the fifth steps, of course, include the evaluation of your 3D construct, both in terms of structure, morphology, functionality, and this will often adopt the same analytical instrumentation that you guys are already using for your existing 2D and 3D models, like fluorescence microscopy, live cell imaging, etc. And finally, you can apply it for your purpose. If it's in vitro disease modeling, you can toss drugs into your tissues to see their behaviors or their death. Number two, you could do in vitro safety and efficacy testing for cosmetic compounds, assessing whether the cells respond as they should according to what is known what is known about certain standard cosmetic compounds. And you could also apply it for in vivo transplantation. And a lot of the real life examples you see downstream later on will talk about just implanting your 3D printed constructs onto mice or onto animals in order to see that they actually continue to live or to regenerate defected parts in those in vivo models. And so this might be an example of what would happen if you were to be using the bioprinting technology for something like cardiac patch engineering. And in your own time, you can take a better look at this with the publication reference at the bottom, because I will be sharing with you these slides at the end of our presentation. So let's now get to the heart of the applications of bioprinting. And slides in the following are actually some summaries or abstracts of actual journal publications that have been made by doctor in vivo users, users of our bioprinting systems. And for more details on these journal publications that draw your attention, let me know or let Sentil know, and we will be happy to send you those journal articles. All right, so first and foremost, I would like to talk about the biomaterial research application using the technology. Biomaterials, as we will now understand from our previous example, these are really the engines of tissue engineering. And the technology of bioprinting has been playing a really big role in characterization and development of novel biomaterials. And in this study by Hanyang University in Korea, the group basically developed a synthetic self-healing hydrogel called OHA GC80H that is basically made from modified forms of hyaluronate and chitosan. And the great thing about this hydrogel is that it forms bonds instantaneously without post-gelation or additional cross-linking processes like temperature, light, and chemicals. And so it's been proving, this is one example that is proving the trend these days to create novel bioinks that can cross-link on their own at room temperature without any additional elements. So as you can see, after getting cut into two pieces, these discs made of this particular self-healing gel reassemble and integrate in less than 10 minutes at room temperature, and it also supported cell viability when experimented with chondrocytes and other cell types that the publication mentions. And so going forward, the group says that this self-healing hydrogel may have a potential in serving as a disc that could go into cartilage regeneration, where this disc serves as the three-dimensional matrix for chondrocytes, and it can gel on its own in the human body. This is an example of soft tissue development. If we step up a level from biomaterials and you include living soft tissue type cells into it. This is a study by Seoul National University, where the group set out to engineer a novel way to regenerate adipose tissues. As we know, in order for regeneration of adipose tissues, some of the conventional materials that have been used as the scaffolds have been PCL, medical grade PCL, very, very commonly used. But the problem that it has been having is that it does not match the mechanical property to adipose tissue. It actually has a almost 1,000 times higher compressive modulus than the adipose tissue. And so the group first developed a PLCL material with a much lower compression modulus to mimic the mechanical environment of adipose tissue. And then they tested whether this particular biomaterial could be printed. And after that, they were able to combine PLCL with adipose-derived ECM Bioink and create these size-specific scaffolds laden with PLCL, ECM, PLCL and ECM, and then they implanted these into mice to show where the vascularization could happen more vividly. And they showed that rather than using PLCL alone, of course, combining PLCL mixed with adipose-derived ECM served as a better scaffold for these vascularization inside the animal models. And this is a very common way for people to test whether the construct that they have made is actually biocompatible and functional inside the living body. Here is another example of soft tissue engineering, especially in particular related to the salivary gland tissue engineering. And in this group, the group worked on developing, I guess you could just focus here. The group developed a way to create a 3D PCL-based scaffold on which they coated with the what they call the niche. It's basically a hydrogel coating technology using hyaluronic acid-based materials, and they tested whether the surface coating on a 3D printed matrix, how that compares with other types of matrices, such as just a regular 4% agarose gel or a cookie cutter PC membrane that is supplied by an outside supplier. And what they have found is that these 3D printed porous scaffolds coated with the niche technology based on catechol conjugated hyaluronic acid yielded a much higher budding levels for these salivary gland microtissues that they could develop. And they also showed that they were functionally viable with the ability to actually see saliva coming out of these 3D cell cultured in vitro tissues. Now, if we move on from the soft tissue models to the next level of models, that may include beating models that beat on their own without any extra electrical pulsations, or these beating models with vascularization. And so we've got examples of using bioprinting to create in vitro models of cardiac tissues, such as what you see here in the images. And there are also examples of creating vascularized cardiac patches by combining these, what they called assembloids. So Assembloids are cell aggregates that are made from different types of cells into balls of hydrogels, and these hydrogels are further printed in order to create these patches. And because you've got both the endothelial cells as well as the tissue-associated stem cells and fibroblasts, they're able to help you create a vascularized model of a tissue rather than just the tissue without the vascularization. And this can be applied not only to tissues like cardiac patches, of course, but to others such as cancer in order to mimic a vascularized cancer model for better drug screening and just physiological studies. Yet another example of cardiac patch engineering using the bioprinting. Here, the key thing was to create very stratified layers of the patch using the elastic properties of alginate, so that the beating of the model could be best preserved. And because of these stratified layers, we could make sure that the cells would not, basically the tissue will not beat in all three dimensions to cause tear of the tissue, but it would do it in a synchronized unidirectional or bidirectional ways. Okay, so I'm running a bit long on time, but let me just go through very quickly I want this presentation really to be able to show you the take-home message, which is that bioprinting is a platform technology with a lot of different application possibilities. And I'm trying to show you these real-life examples that are going from biomaterial development to soft tissue models, beading models, models for drug testing, the printing of drug compounds themselves for personalized therapy, and more. And so please take a look at just the titles of these slides, and maybe if you guys have more questions at the end, I can address them more in detail. So there are also examples of muscle model creation using bioprinting, where you basically print two pillars in the periphery, and you seed cells around it in order to create an in vitro muscle model for novel drug screening. You could also have cancer 3D tumor tissues for the same purpose, 3D vascularized tumor tissues for better models for drug testing. And now if we go on to more of the applications using harder materials, some of these plastics that we are more familiar with, we have an example of creating bone regenerative scaffolds using bioprinting. This is actually an example of the bone regenerative scaffold that have been made by a biotech company called BioOrigin, and they have now commercialized these in the hospitals. More bone regeneration scaffolds. We've also got cartilage regeneration applications, where basically the idea is that in order to battle the limitations of stem cell injections or some type of compound injections that osteoarthritis currently relies on as the only therapies, this offers a regenerative method where you print the 3D printed scaffolds, seed them with the stem cells, and help them to localize onto the knee defect, so that ultimately you could see regeneration of hyaline cartilage rather than just fibrous cartilage as the result. And we've seen these actually in in our own experiments with the Mass General Hospital in Boston, which is one of our partners in the clinical and preclinical studies. And furthermore, we've also got the use of bioprinting to create a channel-like constructs, and applications could range from these, nerve regenerative guidance conduits to perhaps also channels for repairing certain urethral tissues as well. Applications are varied. And finally, not just working with living tissues, but there's been also a lot of research using the bioprinter Dr. in vivo to create personalized drugs with variable solubilities and drugs with variable thicknesses, so that they can be better accommodated by different patients. But another key thing is the digitalization of the information directly onto the pill. So this is a QR coded drug that if you scan, it basically shows compound information. And so what the authors suggest here is that these could basically enhance patient compliance to prescribed drugs, and also become the starting points for the development of smart farm industries. And finally, even microbial metabolism studies are being done with bioprinting. So you don't have to just be working with mammalian cell cultures. You could be working with microbial cells and be adopting a three-dimensional tissue or soft robotic engineering in order to better understand the microbial metabolisms and more. In the end, the ultimate goal of the bioprinting technologies is to offer bedside point-of-care medicine. And so with this picture in mind, ROKIT Healthcare as a company and as a R&D institute has been focusing on translating the technology in the operating room. And these two examples that I will show you are the clinical applications. And we have actually done clinical studies with about 100 patients so far having diabetic foot ulcers. And the idea is that the biomaterials come from the human patient themselves, from the adipose tissue extraction. And the adipose tissue is mechanically micronized into bioinks that can then be printed with the bioprinter according to the defect sizes of the diabetic foot ulcer. As you know, this is just one example. Diabetic foot ulcers tend to have very much irregular shapes, and some of the limitations of conventional treatments are that not only are they either animal derived or they're just composed of single biomaterials, not able to mimic the entire microenvironments for skin regeneration, but they also cannot be aptly customized to the shape and the depth of the ulcer. And that can create a very slow treatment process. And so bioprinting helps to solve that issue by customizing the shape to that defect. And these are some of the results that we have seen just within 2 to 4 weeks. And these are wounds that had not repaired for up to 2 years, or up to 6 months to 2 years. And our ultimate goal is that in order to translate the bioprinting technology as the point of care therapy, we are now working on AI, artificial intelligence based systems, where it becomes an integrated 3D scan print system. So you scan the defect at the hospital, in the clinic, you allow the AI trained program to customize these 3D object files that are specific to the defects, create the flow for the bioprinter to print the regenerative scaffolds using the outlines, the outlining scaffold plus the bioinks from the patient and implanting into the foot. So here is an example of what that looks like. And this is one of our prime applications of bioprinting today at ROKIT Healthcare. This is more of a rough version from several months ago, but I can give you more updates on this later after the seminar. So this is, let me just fast forward. This is basically the idea behind the system. And so again, you are seeing the benefits of automation combined with human autologous biomaterials as the key values for bioprinting. And second, we also have clinical studies now going on with cartilage regeneration, where we are using biomaterials as we're using donor cartilage, microparticles as the biomaterials and combining them with patient-derived adipose tissues as the bioinks in order to create cartilage regenerative scaffolds. And if you guys are interested in that, I can share with you more updates on that later. So with everything that I've said so far, if you guys are curious about some of the features that our systems offer, please feel free to take a look at these next several sections of the slides that I have prepared for you, really trying to depict how these systems work in videos. I'm not going to go over them in detail right now, but just to summarize, this is a system that can accommodate -4 degrees to 350 degrees with a variety of material accommodations from hydrogels to pellets and filaments. We've also got a system called the 4D6 that basically translates the existing key features of the 42 and adds to them this bioreactor system so that you can print inside a controlled chamber of temperature, humidity, and CO2 levels. And of course, the 46 is different from 42, not only because of this bioreactor system, but also because of the high throughput application and as well as this mobile, mobile usability of the software. And so you can work on the system on a tablet or on your phone with the 46. And that also includes the ability to use 6 printheads all at once. And so if we summarize, I hope that I could deliver the message about how bioprinting not only, it doesn't really exist to replace completely the traditional experiments that you have been doing, but it really exists to step them up in terms of automation, in terms of on-demand fabrication capacities, so that you don't rely on perhaps outside sources of less ideal models for your human tissue research. And also bioprinting exists to upgrade the physiological relevance of your models from 2D or from simple 3D to more complex 3D. And so that's just the message I really wanted to drive home. And what a vision that we have in mind is that if these types of bioprinting systems or facilities can be set up all around the world, the value chain that it creates is local biomanufacturing. In the season of COVID-19, we have seen how manufacturing or the supply chain management can really break down across the world with the importance of localities of that manufacturing chain. And I really personally believe that bioprinting can play a role here. And one thing we're working on is we're working with a food printing company, the biggest food printing company in Korea right now, that is now trying to create a food factory using bioprinters to create basically artificial fish meat. But the thing that they said is that bioprinter will play the role of customizing and supplying artificial meat in local regions with food materials sourced from those local regions instead of relying on centralized supplies. So I really thought that that example also speaks to the long-term value creation by bioprinting, and that could be really important for any type of grant winning and project collaborations. And as you can see, this is not just a story, but it's already happening around the world. Some of these centers arising as the bioprinting, biomanufacturing facilities around the world. And at the end of it all, ROKIT Healthcare still stays true to the paradigm of translating this technology for patient benefits. And in our experiences of using the bioprinter to create the right microenvironments for tissues to regenerate, a lot of these experiences have shown us that really human tissues are extremely complex worlds, microcosms that really cannot be recapitulated with any type of three-dimensional fabrication or just single component bioinks or biomaterials or single cell types. It really requires a complex interplay of all of these things. And at the center of that is the ability to really maximize the use of autologous biomaterials instead of relying on outside synthetic or naturally derived biomaterials. So we're spending a lot of time and energy to research into what can be sourced from the patient to become the perfect niche biomaterials to prime the regeneration in the defect area. And these are some of the experiences that we as a company can uniquely provide for the groups that are working with us. So with that, I will end the presentation. Thank you so much for your patience and for your attention throughout. And I hope that this was helpful in at least priming the beginnings of your research in the next levels using 3D and 4D biofabrication. Thank you so much.
Thank you, Dae. All right, now the floor is open for questions.
Thank you so much. Thank you so much, Dae, for the wonderful presentation. So I have a question regarding the machine specifically. So I see there is one the nozzle for the bioprinting or the Bioink, let's say, and the other one is I think mainly for high performance or just polymers. So is there an option we can have two Bioink nozzles? Or is it something that the one which is for the thermoplastics cannot be removed?
Oh, you are talking about the 4D tube, right?
The 4D.
Oh yeah, the 4D2. Yes. Actually, if you guys scan this QR code right now with your phone or something, you'll be able to see our homepage describing this in detail. But I want to show you that this bioprinter is actually able to accommodate on the left interchangeable printheads. So yeah, this left one can accommodate syringe dispenser. the film mixture as well as the hot melt pneumatic dispenser. So you can print with 2 syringes for 2 hydrogels, or you can print with the scaffold laden strategy, where you use the bioplastics on one side and hydrogels on the other.
Okay, understood.
Definitely, yeah. So you can do both soft tissue and hard tissue fabrication with this device as well.
Great. I think I will have more once I go through the specific publications from the 4D2, because the 4D6 haven't yet maybe used by many. I looked at the applications as while you were presenting, and it's definitely diverse. So I will go through some of the applications of my interest, of my research interest, and if I'm sure I will have questions, I'll reach out to you then.
Yeah, of course. Sure.
Anyone else? And here, do you have any questions for Dai? Oh, yeah. Nahyan, you have a question?
Yeah, yes, sir. Thank you.
Yeah, go ahead.
Okay, so thank you for the nice presentation today. I have just one question. While you were showing the application, the cartilage regeneration implants. So for this kind of implants, is kind of clinical experimentation is going on for this, or it's just is still on the experimental phase, not the clinical phase.
For the cartilage regeneration, right?
Yes.
Yeah. We are at the clinical stage. We have just completed a round of clinical studies in Egypt, and we are now going to do it in Korea and other countries in Europe. We are partnered with the Mass General Hospital in Boston, and with them we completed 4 rounds of preclinical studies with beagles up until the end of last year, actually. And we have confirmed that this particular strategy was able to prime hyaline cartilage regeneration rather than the fibrous cartilage, which basically is just like scabs appearing over wounds. And what we want to see is the cartilage that actually has regenerative capacity. And so we have confirmed that in preclinical studies, and that allowed us to go into clinical studies. The publication, I believe the publication for this particular study will come out sometime mid this year. So when that comes out, I'll be happy to share that with you guys.
Thank you.
Okay.
Couple of questions.
Oh, one question.
Yeah. So maybe if I go through the details, I might be able to know. But what are the smallest and largest objects you can print in terms of the dimensions? Right? Is there a limitation on that? Or can I print like 2-inch by 2-inch cardiac patch, or can I print something like 50 microns or 100 micron size?
Yeah. Okay. So definitely between 42 and 46. 46 is the sort of the higher resolution, and 42 is for a bit bigger volumes. So if I talk about the build volumes first, 42 is able to print 10 centimeters by 10 centimeters by 8 centimeters. That's the maximum volume that it could print. 4D6 can print 8 centimeters by 8 centimeters by about 4 centimeters as the maximum volume. So 4D2 may have a bit of advantage in printing larger volumes. On the other hand, 46, it actually has a built-in medical device grade linear robot, and that allows for a much higher precision than some of the existing bioprinters that you may see out in the market. And so 46 is able to print at a resolution of at a layer resolution of basically 1 micron, and the layer thicknesses that you could expect is somewhere around 30 to 50 microns, especially if you're not working with, like very fluidic hydrogels, but let's say you're just printing with PCL, you could print down to about 50 microns. With 42, you can print down to about 100 microns.
Okay, so I mean that's the kind of resolution, and I'm sure if you have different kind of nozzle size, because I think my research and I think I'll just speak on behalf of Chandra. I think we are more towards like a miniaturized tissue patches. So I think that's what maybe Chandra asked, like a patch, which is maybe millimeters or in inches, not centimeters at all. So,
oh yeah, sure.
So we are, let's say, we are the smallest, or maybe the reports which have come out. What's just on the top of your head? What's like the smallest construct or patch that was printed by the 4D2? Can you just say, just on the top of your head, like maybe not an exact number?
The smallest is about 600 microns. So what I've seen is the 42 has been used to dispense 10 microliter volumes of tumor assembloids. So these are patient-derived tumor cells with endothelial and stromal cells in a matrigel. And it was used to print, basically, it's called droplet printing. It was able to print these 3D droplets, and that was ultimately around the size of, if I remember correctly, 600 microns in diameters of the resulting organoid.
Okay.
Yeah.
I think I got an idea about it.
Yeah. But yeah, most of the times people are creating small patches rather than larger ones. We are trying to sort of cross the lines of larger patches because we are talking about treatments, patches for covering a large defect areas. But most people, especially at the research scale, they print at smaller volumes. And with 46, I think you may have seen it at the very beginning. You can print into 96 wells. And you can print into 384 wells as well. So that will give you an estimate of the size of the tissue you can make as well. Yeah.
That is correct. That gives us an idea. Thank you. Thank you.
Yeah, of course. Please feel free to ask any questions that I can address live today, and then any other questions, I'll be able to also share some publications or journals that you guys are interested in. So just send Senthil an email about the specific themes that you're interested in, and I can curate the materials for you as well.
Sure. Okay. If I think if there are no more questions, I think we can thank the speaker once more for this wonderful presentation.
Thank you.
We thoroughly enjoyed it.
Thank you so much.
Senthil, you're on mute if you want to say something.
Sure. Yeah, thank you every one of you for your time and interest. Just before you go, Prabha, what's your timeline like for the acquisition of a new bioprinter?
I'm thinking something. My aim is to have it like that. So this is March, end of April, or so. End of April, beginning of Yeah, end of April, and that's what my aim is.
Okay.
So after this meeting, by today or by tomorrow, I'll reach out to you and we can have a discussion. Okay.
Okay, let's do that. Other than that, if you have any other questions
Sure.
that comes up, do feel free to email me.
Sure.
And
Sure.
I will
And
take care of
I
this.
think I'll share your email also with the others. If anyone is interested, can reach out to you. I'll share it with Chandra for sure, with Muyel for sure. They can circulate it also. If anyone is interested, they will reach out to you.
Sure. And as I mentioned, my company is Ohio based and I live in the Cleveland area. My
Sure.
company is based out of Seoul. And so you will have the best support on a continuing basis. And I assure that you will find in vivo as your best fit for your needs. Plus, ROKIT is a very committed company, and they take good care of their customers. And more than anything, it is the relationship often extends beyond the purchase of the equipment. There is so much happening in this field at such a rapid pace. ROKIT has its own big, robust internal R&D. So a lot of that experience, which is very valuable, also gets transferred to its users. And that is sometimes that is very important to advance,
Sure.
right? Than to try and reinvent the wheel on your own.
Definitely. Definitely. Really appreciate the kind of support which you are providing. We'll definitely look forward to it. Okay.
Wonderful. Chandra, did you have any comments?
I do have, but I just want to go through the material before I open my mouth. Some of these might look too trivial or obvious,
Nothing is
but
trivial.
yeah, but I will read through some of the material.
Okay.
This is the first time I'm seeing this technology, as a presentation. I'm aware of Muil's bioprinter and other things, but
Mm.
I think I will read through and touch base with Prabaha as well as you, and then we'll go from there.
Okay, great. Awesome. And yeah, no problem. Thank you so much. You all have a nice weekend. Then I guess we will let Dai go and
Definitely, and
get back.
definitely a big thank you to Dai that at this point of time in South Korea, you have taken this effort to give us this wonderful presentation, sacrificing your sleep. That's
Tomorrow
really
is Saturday. It's okay.
They are very hardworking people, and we will have
yeah,
some
yeah,
about that some other time. Usually, she is here in the US, but since the
Right.
onset of COVID, I think all that changed, and she kind of based herself in Korea for quite some time now. But
sure.
Dai, you will eventually come back here, right?
Yeah, actually, my family is all in Boston.
Yeah.
Yeah, so I will eventually go back.
So,
Sure.
I'm enjoying my time in Korea too.
fun. All
Sure.
right. Thank you so
Okay.
much,
Yeah.
everyone.
Thank
Have
you.
a wonderful day and a
Thank you.
nice weekend. We'll be in touch.
Have a
Sure.
good
Okay.
Friday.
Bye
Have a
bye.
good
Bye bye,
day.
everyone.
Bye.
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 For making
Bye.
the opportunity.
Bye.
Bye.
See you.